In this video, we're going to learn about the transport of materials across membranes. So materials need to be able to pass across membranes. So we need to be able to get nutrients into the cell, water into the cell, and then waste needs to be removed from the cell. So they have to have a constant flow of materials into and out of the cell. Cell membranes are said to be selectively permeable, so not everything can just pass through or exit at will. There are controls and measures set in place. And there's several methods that allow for the transport of materials across a membrane. Some of them require energy, and those are referred to as active transport. And then some of them do not require energy, and that would be passive transport. The first type of transport across membranes would be diffusion, and this is the one I think most people are more familiar with. So that's the passive movement of molecules from an area of high concentration, so an area where there's a lot of particles congregated together, to an area of low concentration where there's not so many particles. So they'll move across the membrane, and you can see here in our diagram over time. Imagine we had this cell membrane, and then all the particles of whatever substance this is are congregated on one side of the cell membrane. And then over time, those particles are going to keep bumping into each other and pushing on each other, and eventually some of them are going to be pushed through the cell membrane until eventually you end up with a balance of particles on both sides of the membrane. And this is said to be equally balanced. So a variety of measures will affect the rate of diffusion. If we increase the temperature, the particles will be moving faster, so they're more likely to bump into each other, and so they're more likely to spread out in the system across the membrane. If you increase the concentration of either of the particle, you'll end up with more particles bumping into each other, so a faster rate of diffusion. And if you increase the pressure, there's more force pushing on the particles, so they will transfer across the membrane faster than if there were a lower pressure. So all of these things will increase the rate of diffusion. If you think of a real life example of diffusion, if you were making a cup of tea and you had a tea bag and you placed that tea bag into your cup of hot water, over time, the tea particles will seep out of the tea bag and spread evenly through the entire cup of tea. And then you end up with a nice, yummy cup of tea. Another example of diffusion might be if you sprayed a spritz of perfume in an area. The person further away won't notice it right away because we have to wait for those particles to disperse and diffuse throughout the entire room. So facilitated diffusion is the passive movement of molecules across a protein channel. So it's just like regular diffusion, except now the particles have to go through this channel. And they're still going with the concentration gradient, so they're going from an area of high concentration to an area of low concentration. But the difference here is there is something preventing them from being able to just transfer through the membrane itself. So they need to go through a channel. There's no energy required, it's still passive, but they are moving through a channel. Our third type of transport across membranes would be osmosis. And in osmosis, this is a diffusion of water across the cell membrane to balance the concentration of a substance across it. So this occurs when a substance is unable to move itself across the membrane. So then water goes across the membrane to dilute the solution on one side or the other. So cells can be in three different types of environments. Isotonic environment, that's what cells want to have. They want to have isotonic conditions. So iso means the same. So they want to have the same concentration of solute on the inside of the cell as on the outside of the cell. So if they don't have that, if they're in one of those other two environments, they will make measures, move water across the membrane to balance out that concentration and achieve the isotonic conditions. So the other type of environment would be a hypertonic environment. So hyper, so it has a lot more solute in the environment than in the cell. And then hypotonic means that there's a lot less of the solute in the environment than in the cell. We'll spend some time talking about each of those. So our first type of environment is isotonic. So the concentration of the solute is equal both inside and outside of the cell. So there's no net movement of water in this case. So there's two different examples of cells here. This would be a red blood cell, and this would be a plant cell. So remember, animal cells don't have a cell wall, so they can actually grow and shrink. So they're more flexible, whereas a plant cell has a cell wall, so you won't see great changes in the size or shape of a plant cell, but you will certainly in the shape of an animal cell. So in this case, because the concentration is the same inside and outside of the cell, for every water molecule that enters the cell, one will leave. So there is no net movement of water. So the concentration stays the same, and the balance of water is the same across the membrane. So a real-life application of an isotonic environment would be in medical procedures. So we would bathe tissues or organs in a medical procedure with something with the same osmolarity as the cytoplasm or the surroundings to prevent osmosis. You don't want your cells to suddenly start losing water or gaining water and then bursting. So we have to make sure that whatever the solution is, that it's the same osmolarity as the surrounding cells. Now in a hypertonic environment, the concentration of solute in the cell is less than the concentration in the surrounding environment. So if you take a look at this, there's a lot more particles outside of the cell than inside of the cell. So because of that, water will start leaving the cell to help balance out that concentration, so to dilute the solution outside the cell. So if water is leaving the cell, if it's an animal cell, will shrivel right up, and then it will be unrecognizable. In a plant cell, what happens is the water leaves, the cell wall still retains its shape, but the cell is said to plasmolize, so basically it dehydrates from within. So the vacuole would lose water, everything would just kind of condense in on itself. Now in a hypotonic environment, the concentration of solute in the cell is higher than the surroundings. So here's our plant cell, and here's our animal cell. And in this case, water will start moving into the cell to balance the concentration of solute across the membrane. So in this case, we kind of show that the cell is starting to kind of swell a little bit and press on the cell wall. As water is getting in, the cell really can't get much bigger. And in this case, in the red blood cell, water is entering, and eventually it'll get so big it won't be that biconcave shape anymore. It'll actually become like a giant inflatable ball, and eventually it would actually break and burst or lyse. So active transport does use energy, and this is when a cell uses energy to move substances against the concentration gradient. So they're going against the flow. Usually with the other types, everything is going with the flow, so with the concentration gradient, whereas with active transport, you're pumping things against the flow. So you actually have to push it across the membrane using energy. So cells will use ion pumps or endocytosis or exocytosis to get things across the membrane, and that all requires energy. So you can see here in this diagram, there's a particle trying to get through the membrane. So ion pumps are like protein channels, but they use energy to function. Endocytosis, if you don't remember, is when the cell wraps the cell membrane around a substance to bring it into the cell. So an example of that would be a white blood cell engulfing an invader to digest it and protect the body against infection. Exocytosis is when a small vesicle or sac travels to the cell membrane and then fuses with the cell membrane and expels its contents out of the cell. So that's when the Golgi apparatus prepares proteins to be sent out of the cell. These all use energy because they're active transport.